Apoptosis. So, what is apoptosis? It is basically a coordinated cell death and responsible for mediating cell death in various physiological and pathological processes. So, here is a list of some physiological processes. Like first, it is organized cell destruction in reshaping of tissue during the development of embryo. Then physiologic shrinkage of cells in any hormone dependent tissues like endometrial shedding जो होती है, so इसमें भी physiologic shrinkage होता है cells का. Then normal cell destruction followed by any like proliferation in intestinal epithelium. Then shrinkage of thymus. Now some pathologic processes are like cell death in tumors. Tumors में जो cell death होती है, that is, and then by cytotoxic T cells. In immune mechanisms like any like जो graft versus host reaction होता है, so that is cytotoxic T cells उसमें involve होते हैं. Depletion of CD4 plus T cells in AIDS, then viral infections में जो cell death होती है, and in degenerated tissues of CNS. Now, some of the morphological features are like single or small cluster of cells. Then it will be round. Apoptosis may it will be round to oval in shape, shrunken with eosinophilic cytoplasm. Then there is pycnosis. What is pycnosis? It is nuclear chromatin जो होता है, it is condensed under nuclear membrane. So this is pycnosis. Apop Apoptotic bodies are formed in this, and no acute inflammation is seen around apoptosis. Then some of the biological changes, if we see, so this is basically proteolysis of cytoskeletal proteins. Biological changes में proteolysis हो जाता है, जो भी cytoskeletal proteins होते हैं उनका. फिर प्रोटीन प्रोटीन क्रॉस लिंकेज जो होती है, that is also there in biological processes. Then there is fragmentation of chromatin after pycnosis. हमने अभी जो pycnosis पढ़ा उसके बाद में there is fragmentation होता है chromatin का. Then there is appearance of thrombospondin on the outer surface. Outer जो surface है वहाँ पे thrombospondin का appearance हमें देखने को मिलता है. Then what are the techniques to identify and count the apoptotic cells? So there are some of these techniques by which you can count the number of apoptotic cells and you can identify. So first is like staining of chromatin condensation by hematoxylic stain. जो हमने hematoxylic stain के बारे में already we know. So staining करते हैं हम chromatin की. Then fluorescent stain with actinic orange dye and flow cytometry, okay, to see any rapid cell shrinkage. So flow cytometry also we do. Then last it comes your gel electrophoresis. Then molecular mechanisms of apoptosis. So what are the molecular mechanisms there? First is like initiators क्या होंगे apoptosis के? There is absence of certain hormones, growth factors and cytokines. Then we have cell death. इसमें final phase आता है, that is the final phase is that mitochondrial pathway जो है, it is going to activate the caspase and they will cause skeletal damage or any disruption of ER और mitochondria का भी damage हो सकता है. Then after that occurs phagocytosis.